Thank you for the invitation for me to present my project. So I changed my title a little bit. Sorry. I tried to include more recent data. So now my title is a synergistic effect between iron chelators and DNA damaging agents as a kind of a group of treatments in colorectal cancer. So I'm from Biochemistry. So this project is driven by this hardworking, talented graduate student, Kim, who is sitting here. This picture was taken before the pandemic, so we haven't really have a group lunch together for a long time. Hopefully we can do that again soon. And then some other lab members participate in this project. Luke, which is a Ph.D. grad student. Jolly is a postdoc. Daniel is a former postdoc in my lab. And have some collaborators from UNM. So David Martin is a pathologist. Henry Lin is at the VA. Olga was at the Division of Gastroenterology. So Mike Dam, Justin, and Yatrig are all from my old institute, University of Michigan. So this kind of project is kind of from it's a deviate from other projects funded by NIGMS and American Cancer Society, and our cancer center provided some matching funding for grad student Kim. So the real innovation for my project is quite simple. This is basically the take-home message for you. It's a combination of iron chelators and DNA damaging agents can cause a synergistic effect by inducing DNA damage response. And this response will cause more cell death, and through this can reduce colon tumor cell growth in both in vitro cell culture and in vivo in animal models. So why we started this project? Because based on cancer statistics from American Cancer Society, so 1 in 2 men and 1 in 3 women will be affected by cancer during their whole lifetime. And among them, colon cancer is really the third most prevalent cancer in both men and women in the United States. So there's an urgent need to develop really effective therapeutics, because even though we have studied colonoscopy trying to detect early diagnosis, but still no real effective and safe therapies for advanced diseases. So accumulating data, including our own, we think iron metabolism plays an important role in colon cancer. So colon cancer cells are addicted to high iron diet, but not normal cells. So this could be really provide a possible safe and effective strategy to treat colon cancer. So this is a review paper we published this year, trying to summarize how we can target iron metabolism. But still, the iron metabolism is really a complex system. So we still don't know how exactly to target this to treat cancer. So there was a paper published in Blood many years ago. They showed that use a metachelator called tachipyridine. So this tachipyridine at a low dose, 7.5 micromolar, can sensitize colorectal cancer cell line HCT116 to irradiation. So, but this even treated at a high dose, 25 or 30 micromolar, it won't affect normal fibroblast cells. They think this mechanism was through this ATR phosphor Chk1 DNA damage signaling pathway. So this will activate this DNA damage response, causes G2 arrest. So for this DNA damage response inhibitors, because you know in 2014 the first PARP inhibitor was approved by FDA. So now this has really gained lots of attention. But this DNA damage response inhibitor, including what I mentioned, ATR inhibitor and Chk1 inhibitor, their monotherapy often has very limited efficacy for cancer treatment. So for the industry, Merck is one of the leading company who has invested a lot in this DNA damage response signaling pathway. They have more than 10 clinical trials ongoing in various tumor types. It just this spring, so they had made some advance in one of their ATR inhibitor. So they basically published a paper in Cancer Cell. So they did a drug screening and found this this ATR inhibitor as well as Chk1 inhibitor can synergize with top isomerase inhibitors, then cause replication stress. Then they think this is effective in animal models as well. They are doing clinical trials. They think this is a promising drug. They may advance to registration. So this is an example for ATR inhibitor. So for the Chk1 inhibitor, there are more kind of problems. So Lilly put lots of efforts on one of the Chk1 inhibitor, preservative. So, but they abandoned this development in 2019, even though there's lots of high-profile, high-impact papers published, and they also used in clinical trials. I was trying to figure out the reason, but I couldn't find out why exactly they abandoned this drug. There was one only Chk1 inhibitor, SRA737, is currently under development by a small company called Sierra Oncology. So they are doing just finished phase 1-2 trials of this drug in monotherapy and in combination. So recent years. So it was last year they published a paper in Cancer Research. They showed this Chk1 inhibitor SRA737 had some synergistic effect in several lung cancer cell lines as well as colorectal cancer cell lines. You can see this different cell line, they have a synergistic effect. This synergistic effect with this DNA polymerase inhibitor called Ph.D. choline. But unfortunately, this Ph.D. choline is very toxic it's impossible for use in vivo. So I think this is really comes to connects our research. So we found our iron chelators can effectively inhibit this DNA polymerase in vitro. So we think these iron chelators may synergize with these ATR Chk1 inhibitors as well. Currently, there are 3 FDA-approved iron chelators, including deferoxamine, deferoxyrox, and deferpron. We did many in vitro studies show, yes, indeed, these iron chelators can synergize with ATR inhibitor as well as Chk1 inhibitors. So then we were trying to get a more broader application, because this 5U, which is a standard static agent, is widely used to treat colon cancer. However, so main concern is this chemotherapeutic drug has systemic toxicity because you have to use a really high dose. Since its pharmacokinetics, its profile wasn't that great. It's poorly absorbed and has a low plasma half-life. So we think iron chelators may synergize with low dose of 5U. This would be enhance the efficacy and reduce the toxicity. Also, we think iron chelators may synergize with irradiation based on that paper I showed you earlier. So I just show a piece of data to show this really the need or the market potential. So this figure, you can see right now the colorectal cancer 2020, as I mentioned earlier, is the third most prevalent cancer, but probably 10 years later, it will surpass lung cancer, and 20 years later, it will surpass breast cancer. It will become to the first place among all cancers. So you can imagine how big the market will be. So also for our data, I just show one slide, show you this is a DFX, deferoxyrax, at a really low dose, 10 micromolar, and this 5U is also very low dose, 10 micromolar. So they single use, they can kill some cancer. This MC38, it's a murine colon cancer cells, so they can kill probably 60, 70, but when they treat together, it can get lower than 50. And this gamma H2AX, which is a DNA damage marker, we can see clearly this combination treatment can synergize this DNA damage effects. Also, very importantly, this in vitro combination can work in vivo as well. So we used probably 50 milligram per kg of this deferoxyrax, and I think it's 1 milligram per kg of this 5U. So you can still see very robust anti-tumor effect in xenograft models. So this project current status, together with some other data, I have already submitted a manuscript at countries at a review at a gastroenterology. Hopefully it will get back soon, maybe later this month. I also submitted a grant to DOD, the impact award. And also we are trying to develop some collaboration, international collaboration with the Egypt group. We are this 5U, lots of times in clinic, it's a combination regime called a 4-FOX. So it's a folinic acid, 5U, and oxaliplatin. So we are thinking this therefore it may also can potentiate this clinical used regime. We are trying to manufacture nanoparticles. Hopefully this grant will be submitted soon. And for future plan, may want to think about what would be the best combination, because there's lots of choice here, and trying to see confirm the toxicity. And we have patient-derived organoids, which is one of the most state-of-the-art techniques. And hopefully, eventually, we can do some clinical trials for this regime. And that's all what I want to say today. Thank you.